In my presentation today, I am going to be talking about wood frogs. The scientific name of wood frogs is Lithobates sylvaticus. Wood frogs are a class of amphibia of phylum Chordata. They are a species of frog found only in North America. These frogs primarily live in the woodlands and forests of the northern United States and throughout Canada. Their diet consists of small forest floor invertebrates like insects, slugs, and worms. During the fall, in preparation for winter time, the wood frogs accumulate high levels of glycogen, which will later be converted to glucose. Glucose is a powerful chiroprotectant and serves to protect cells from desiccation once the wood frogs begin to freeze. Once temperatures drop close to the wood frog's freezing point, it will find a spot near to a river or other body of fresh water to settle down for hibernation. The wood frog burrows itself under leaves by laterally rotating its body in the soil. As soon as it begins to freeze, the frog's metabolism and heart slows and eventually stops completely. All other organs stop functioning as well. The frog no longer uses oxygen, and for all intents and purposes, it appears to be dead when it's in this dormant state. However, amazingly enough, when spring finally arrives, bringing with it warmer temperatures, the wood frog begins to thaw and its heartbeat returns. This is an amazing phenomenon, and it is no wonder that scientists want to know more. Once the wood frog thaws, the wood frog soon makes its way to the nearby body of fresh water to breed and lay eggs. Wood frogs actually prefer vernal ponds rather than lakes or larger bodies of water. Vernal ponds are small, temporary ponds arising from the spring snowmelt. The reason that they prefer these are because lakes and bigger bodies of water harbor fish and other predators that might eat their eggs and tadpoles. This is also why wood frogs have an unusually early mating season. The window for their tadpoles to grow and properly metamorphose into juveniles is quite small since the ponds are only temporary and will dry up as summer approaches. This is just a little bit of background information about the wood frog. Now we're going to move on to talk about what their role is within the ecosystem in which they live, and then we're going to talk about what benefits we can gain from learning about them. Organisms within a food web balance each other's populations and stabilize an ecosystem, and the wood frog is no exception. Wood frogs have various predators and thus are an important source of food for many animals in their ecosystem. Some of their predators include garter snakes, ribbon snakes, herons, raccoons, and skunks, to name a few. Wood frogs also feed on many terrestrial and aquatic invertebrates as a part of their diet and therefore help control insect populations. Another crucial role of the wood frog from a human biologist point of view is their ability to indicate environmental health. Frogs have very thin and permeable skin in order to be able to absorb water and breathe on land. This is because their lungs are not sufficiently developed to obtain all the oxygen they need. This highly permeable skin makes it easier to not just absorb air and water, but also bacteria, chemicals, and other toxins in the environment. So if a bunch of frogs in a local ecosystem start getting sick and dying, then that should be an indication that there is something more going on, such as high levels of a chemical pollutant or something of that nature. There are additional applications of studying wood frogs, especially within the medical field. The ability of wood frogs to freeze, completely stop their hearts and organs from functioning for many months at a time, and then recover completely is an area that is full of practical applications. One of which is the preservation of organs for transplant. There are many people who need an organ transplant in order to stay alive. But the problem is that organs die pretty quickly unless they are frozen after being harvested. But when doctors thaw the organs, they are often damaged due to cracking and the thawing process in general. So studying how wood frogs are able to freeze and then unfreeze their organs without fatal damage could potentially lead to new technology that could allow for long-term storage of organs, which would enable more people to get the transplants they need. Other applications include cryosurgery, which uses extremely cold temperatures for the removal of cancer cells. Glucose management in diabetics is another one. Wood frogs are able to accumulate very high levels of glucose in their bodies without damage or death. And finally, tissue damage often results when someone suffers a stroke or heart attack when the blood returns to an area after being absent a while. This happens to wood frogs as well when its blood starts to flow again in the spring. So understanding how damage is prevented could be useful. Now that I've covered the basics about wood frogs and why biologists are interested in studying them, let's take a closer look at an article published in 2014 covering many years of fieldwork and observations about this unique species. Prior to this study, most scientific studies of wood frogs focused on the Midwestern United States and Southern Canadian populations, which are near the southern limits of the wood frog range. The authors of this article, however, wanted to observe wood frogs living near the northern limits of their distribution, so they studied wood frogs in Alaska. Alaska, being as cold and harsh as it is, only hosts 2 native species of frog, one of which, of course, is the wood frog. This group of scientists set out to observe a number of different things. They wanted to measure the frog's duration of freezing, so how many consecutive days they were frozen and at what temperature, their rates of survival over multiple winters, their behavioral and physiological responses. They wanted to measure levels of glucose accumulation in tissues. And finally, they wanted to test for the presence of AFGL, which stands for antifreeze glycolipids. Before this study, it was unknown whether wood frogs contain AFGLs. Their methodology included both field and laboratory studies. In the field, they tagged 18 free-living wood frogs over the course of 2 winters, placed wire mesh cages over each frog to prevent relocation, and set up temperature probes to record the temperature every 5 minutes during the winter until they reemerged in spring. The authors also set up an outdoor enclosure over the course of 2 winters, which was designed to mimic real conditions. They also froze wood frogs in the lab and had a control group in the lab that remained unfrozen. The free-living wood frogs, which were studied during the winters of 2001 and 2002, had 100% survival. They were frozen for an average of 193 days, but the number ranged from 175 all the way to 218 days of being frozen. Between October and May, the minimum temperature experienced by the free-living wood frogs was negative 18.1 degrees Celsius. This chart on the left shows the average daily temperatures for the soil and air during these winters. The frogs in the outdoor enclosures were studied during the winters of 2011 and 2012. The minimum temperature they experienced in 2011 was negative 22 degrees Celsius, and in 2012 was negative 17.5. Glucose concentrations were measured in the frog's leg muscles, heart, and liver. The amount of glucose in free-living wood frogs and wood frogs in outdoor enclosures were pretty much the same. There was no significant difference, so the authors decided to combine the data from both and group it into one category notated as field in this chart, in orange. The field wood frogs had glucose concentrations that were significantly higher than those in laboratory frozen frogs. They hypothesized that this was due to the freeze-thaw cycles that field wood frogs undergo. Under natural conditions during the fall, wood frogs freeze at night and then thaw slightly during the day when it gets a little warmer. This process leads to increased glucose accumulation in tissues. The laboratory frozen frogs, in contrast, were merely frozen once, and so only made glucose that one time, and that was that. They also found that laboratory and field frozen wood frogs had higher glucose than unfrozen control wood frogs, which makes sense because they don't need glucose as a cryoprotectant if they are not being frozen. As mentioned earlier, the scientists also wanted to test for the presence of AFGLs, antifreeze glycolipids. To do this, they first tested for TH activity. TH stands for thermal hysteresis and indicates the presence of either antifreeze protein or antifreeze glycolipids. They found that there was high TH activity in the frog's skeletal muscles and organs, but minimal activity in their skin. Next, they wanted to determine whether the TH activity was due to an antifreeze protein or a glycolipid. They did this first by treating the samples with trypsin, which demonstrated that the activity was not due to a protein. Then they treated the samples with xylanase to demonstrate that antifreeze glycolipids were likely responsible for the TH activity. After this, they compared their frog samples to a sample of well-studied freeze-tolerant beetle. They did this because the beetle had already been shown to use antifreeze glycolipids. They compared the region of antifreeze glycolipids from the beetle to the same region in the frogs. They found that there was good correspondence between the two spectra and were able to conclude that the frog sample was chemically similar to the beetle one in terms of AFGL. This 2014 study was the first to report on the presence of antifreeze glycolipids in wood frogs. Scientists have known about antifreeze proteins, but the discovery and study of antifreeze glycolipids is still relatively new and their function unknown. Like antifreeze proteins, glycolipids are thought to have a role in reducing the freezing point of organisms and in controlling ice formation within their bodies. Again, understanding the exact mechanisms might help us with the practical applications, like successfully freezing and thawing human organs without damaging them. The results of this study also showed that wood frog freeze tolerance is more extreme than previously established. Before this study, lower lethal temperatures of Alaskan wood frogs were reported as negative 16 degrees Celsius. This study further extended this minimum to negative 18.1 degrees Celsius under natural conditions. Most likely, wood frogs can survive still lower temperatures than this, because where they live, temperatures regularly get below negative 20 degrees under the snow. Duration of freezing survival in wood frogs in Alaska was much longer than reported from other studies, with temperatures remaining below the freezing point for up to 218 days, which is over 7 months, with 100% survival. Wood frogs in Alaska had enhanced freeze tolerance compared to wood frogs in more southern regions. This is likely due to the freeze-thaw cycles they undergo during autumn, as I talked about before. Alaskan wood frogs amass higher amounts of glycogen in preparation for winter. Then they undergo these freeze-thaw cycles when they freeze during the night but thaw slightly during the day when temperatures warm up a little. Each time they freeze, they convert more glycogen into glucose. But during the day, it's not warm enough or long enough for the frogs to reclaim much of the glucose they've produced. The authors also concluded that freezing tolerance in wood frogs was likely enhanced because of the presence of antifreeze glycolipids in their membranes and tissues. They appear to have an AFGL similar to those described in other freeze-tolerant species like the beetles. The authors also suggested that further study would be necessary to understand the exact mechanism and function of the AFGLs. All in all, this article suggested that the extreme in freeze tolerance of wood frogs in Alaska, compared with more southern populations, was due to differences in the climate and weather patterns, leading to freeze-thaw cycles and higher glucose levels. However, this might not necessarily be the whole story. Maybe the wood frogs living further north underwent evolutionary change and simply have different genetics than those living down south. This hypothesis could be a good jumping-off point for a follow-up experiment, which could utilize genetics to compare the DNA of northern and southern wood frogs. Thank you for listening.